Our next talk in the illustrious fat session is from Richard Dohmen, who's a scientist at Mosa since 2019 as well. And he is famous for Fibro-Adipogenic precursors, and will tell us some more about them now.
Thank you, Freya, for that nice introduction, and thank you, organizers, for allowing me here to present today. So my name is Richard. I'm indeed a PhD candidate and scientist at Mosa in the cell biology team, and I will be talking about some improvements that we made to our animal-free culture of the muscle-derived Fibro-Adipogenic progenitor cell. So as you already heard this morning from a really nice presentation from Sophie Hubalek, and also in the really nice presentation of Johanna Melke this afternoon, we at Mosa use the Fibro-Adipogenic progenitor cell, and that's mostly because we can obtain it concurrently with satellite cells, as the Fibro-Adipogenic progenitor cell actually is responsible for all the, all the marbling that we see here in the steak. And as you also heard from Johanna, well, she mentioned it casually, we published an article about that last year, highlighting some of the advantages of these FAPs and why they make for such an excellent cell candidate for cultured fat. But actually, also in that article, we have two slight problems. One of them, yes, they efficiently proliferate, but that proliferation that occurred in serum. And secondly, yes, our FAPs, they differentiate quite nicely into mature adipocytes. However, over time, with more population doublings, they basically lose their lipid volume per cell. So the older the cells, the less fat we have, sadly. So first things first, can our FAPs be grown in serum-free medium? Well, I think Johanna already gave that away. But so to test that, we used the same formulation that was devised for satellite cells by Anna Kolkman in 2022. And this is the formulation. And from this formulation, we already removed fibronectin because it's way too expensive. And we did, however, test fibronectin as a valid coating option for FAPs, by the way. So using this formulation, we basically compared our FAPs to serum-containing medium, and we found that, yes, our FAPs do grow in serum-free medium, however, albeit a little bit slower compared to the serum-containing medium. So as I said, this medium was specifically designed for satellite cells. So now we wanted to tailor it a bit more towards the FAPs. And I'm specifically going to mention the work that I did regarding all these supplements that I listed here. So these are mainly the growth factors and also the ITSX complex. So first things first, indeed, the growth factors that I highlight here. So what we basically did is we cultured our FAPs in medium that contained all the growth factors, no growth factors, and all the growth factors that are listed down here. And what we saw, well, we did that for 4 days. Why doesn't it bugger off? So we cultured these cells for 4 days, and we represented it here as the PDs per day. And as you can see, only FGF, IL-6, and PDGF alone have like a positive, they stimulate the proliferation of FAPs. And it was actually quite nicely that it corroborated also our RNA sequencing data, given that FAPs, they express the PDGFR alpha receptor quite highly compared to satellite cells. And also conversely, satellite cells, they actually express the HGF receptor quite highly, whereas FAPs barely don't. So that nicely corroborates also this data. And I have to mention that this data is also irrespective of the concentration. So we tested a logarithmic scale of the concentrations, and only in FGF in purple, IL-6 in green, and PDGF-BB in blue have like a dose response curve. So the higher the concentration, the more effect proliferation you see. Next, we obviously tested several of these combinations. You can find the combinations on the bottom. But what I would like to draw your attention to is that these 4 combinations, and what all these combinations have in common is that they contain FGF, IL-6, and PDGF. And all these 4 combinations go at a similar rate as the all growth factors. So that leaves the question, can we remove HGF, VEGF, and IGF? Well, first focusing on HGF and VEGF. Yeah, basically yes. So we cultured the cells without VEGF, HGF, and the combination, and we saw that there was no difference at all in the proliferation. So that's shown here on the left with the cumulative population doublings, and the graph in the middle shows the PDs per day, so the growth rate. There was no significant difference, nor was there in the graph on the right, which shows the differentiation. So they're out. Now, obviously, that leaves the question, what does that do to the costs? Well, so highlighting, I'm only going to highlight here the cost for the supplements as a percentage. So if we then look at the supplements minus the fibronectin and we remove these two growth factors, we already see an 11% reduction. And I have to add here that if you take the entire medium, if you combine all the medium, the cost of the medium, it still had quite a high impact. It was still quite significant, only removing these two, although it might not seem as much. So next, we obviously went on to IGF-1. As I said, it also didn't really have a proliferative effect. However, in our medium, our medium already contained insulin, which is in the ITSX supplement, which stands for insulin, transferrin, selenium, and ethanolamine. So first we had to get insulin out of that mixture. So we bought all these components separately, which already reduces the price, of course. And whilst doing that, we actually already noticed that we can grow our cells without transferrin. So from ITSX, it went to ISCM, I suppose. And thereby already removing 40% of our supplements cost. It's quite a significant reduction. So next, finally, we could do the test with insulin and IGF-1. And as you can see, there was no significant difference in either of them. So both insulin and IGF-1 could proliferate these cells. And also in terms of the differentiation, there was no difference at all. However, if we remove both insulin and IGF, we do see that there is a reduced growth rate, so reduced proliferation, but an increase in differentiation, which is quite an interesting effect. But yeah, as I said, so this data suggests that either insulin or IGF-1 is sufficient for proliferation and differentiation of the FAPs. So in theory, we could already remove the entire insulin, transferrin, selenium, and ethanolamine supplement. However, yeah, well, of course, the actual decision will be based on the biological pathway. So which one of these pathways is actually more important, and also the efficiency of either of these, because they can bind on each other's receptors. So the efficiency of these bindings, and the concentration, and obviously also the cost. So at the moment, we're still looking into that a bit more. So now that we have a medium that we kind of like, we obviously ask the question, all right, what is the proliferative and differentiation capacity of our FAPs in this medium? And to do that, we did a long-term passaging and differentiation experiment. So here you can see the average cumulative population doublings of 11 isolations over the course of almost 2 months. Well, it's more than 2 months. And as you can see, they already almost reach 40 population doublings. And here you can see all the isolations separately. And as you can see, some isolations tend to grow a bit quicker than others, but overall they keep quite a nice trend. We also extended the proliferation for roughly 1 month longer for 4 of these isolations, and we saw that they can actually reach quite nice high limits relevant for cultured meat. So between 40 and 50 population doublings, and I think Andrew already mentioned the Hayflick limit. It's where they basically ceased to grow. And over the course of the entire experiment, our PDs per day were roughly between 0.5 and 0.6 population doublings per day. So next, of course, the differentiation potential of our FAPs. So here on the y-axis, you can see the lipid volume per cell, and on the x-axis, the passages with underneath the average cumulative population doublings of all those 11 population isolations combined. And each dot in this graph basically represents one isolation, as you can see, we have quite some iso-to-iso differentiation differences, as Johanna also already mentioned. But what I would like to draw your attention to is the drop that we have from P9 to P11, basically between population doubling 22 and 26 is where our FAPs start to lose their adipogenic differentiation potential. So that obviously raises the question, can compounds actually extend this adipogenic differentiation? Because our FAPs, they can grow almost up to 40 to 50 population doublings, so it would be nice if we have also differentiation up to 40 or 50 population doublings. So the same graph, so we're basically looking for compounds that can extend the lipid volume per cell at any given population doubling, or obviously compounds that can maintain the lipid volume per cell over time. Now, a little bit about our compound screen. So we basically, as also Johanna already mentioned, she's mentioned a lot, we do our compound screen at a 2D surface by using the high content analyzer and BODAP. So BODAP stains neutral lipids, and what the high content analyzer basically does is assigns the BODAP positivity to the nearest nuclei, and thereby spitting out a percentage of differentiating cells. And this is just a representation of how it looks like. So we tested several compounds ranging from the TGF-beta pathway to MAP kinases inhibitors, and also some of the hedgehog pathway. But what I really would like to draw your attention to is the one now on the right, LCKSL, Loxel, as we called it, which was quite surprising because it was actually supposed to inhibit the differentiation, but it basically does the opposite. So we had more positive, so there was more differentiation. But then we had a look at our solvent control, and we actually saw that, yeah, we had as high percentage of body positivity there, and then, which was quite surprising. And this solvent control is DMSO. Who knew? Indeed. So next we obviously did so we differentiated, we proliferated the cells in a concentrations range. So we exposed them to concentrations ranging from 1 on 1,000 to 1 on 100. And with an increasing concentration of DMSO, we also saw an increase in differentiating FAPs. So next we did a long-term proliferation and differentiation experiment. And yes, DMSO is quite toxic, so if you increase the DMSO concentrations, that's also quite detrimental to the proliferation of the FAPs. So 1 on 100 wasn't really growing that nicely. But overall, 1 on 300 did quite well. And if we then look at the proliferation, the differentiation, so again on the x-axis we can see the passages, and underneath the cumulative population doublings per passage. We can say that, well, this isolation is quite well at keeping its stemness, its differentiation capacity, but at any given passage, at any given population doubling, DMSO 1 in 300 was able to, so it's significantly more fat at any given point. So at this moment, we're not really sure how DMSO works. There's not a lot of unambiguous data about the cellular effects of DMSO. There is some suggestions that it has an epigenetic effect. What we do know is that we have FAP heterogeneity. So we recently did a single-cell RNA sequencing where we saw 5 different subpopulations within our FAPs, and these over the course of passaging, these either appear or disappear, or they change in their in size. So it's quite likely that, well, it's not quite likely. It might be that DMSO has an effect here on the FAP heterogeneity, on either the presence or the size of these subclusters. And with that, some quick take-home messages. I see I have less than 1 minute. So we reduced the cost of our serum-free growth medium, and we still maintained a similar performance. We determined the proliferative and differentiation limit of our FAPs, and we extended also this limit using DMSO. And in the future, we obviously want to understand how DMSO works, how it increases the adipogenic differentiation, But also the decreased proliferation and increased differentiation, what we saw with the insulin and IGF, is quite interesting. And eventually we would like to translate these findings to upscaled cultures. And with that, I would like to thank, obviously, all these people, but especially Professor Mark Bost, of course, and also my team leader, Josh, and you. Thank you.
Okay, guys, time for one question. Oh, it's always so hard. Where's the box? He looks really excited.
Yeah. No, it's just really interesting about the DMSO because we use it for making hepatoblasts to drive differentiation. But there was a paper from Doug Melton's group about, I think, 8-9 years ago, stating that DMSO pushes cells, especially stem cells, into G1. So it could be a cell cycle thing with
nice.
the DMSO.
All right.
So I'll forward the reference to you.
Yes, please. Thank you.
That's so interesting. Familiar compounds for unfamiliar uses. Thanks so much. Really, thank you so much.
Okay.
Really excited to...